Hello, the title of my talk is the Zinc-dependent transcriptional regulation of miR-224 confers ZIP4-induced apoptosis resistance in pancreatic cancer. This is my poster. Our lab have been working on the function of the Zinc transporter called ZIP4. We found ZIP4 was significantly increased in pancreatic cancer compared to the normal pancreatic tissue. And ZIP4 play a very important role in pancreatic cancer tumor progression and development. Recently, we found that microRNA called miR-224 is negatively correlated with ZIP4 in pancreatic cancer cell lines and tumors. So when they have high ZIP4, the miR-224 is low, while knocking down ZIP4 lead to increased miR-224. Furthermore, we validated this finding with the human PDAC tissue sections. We found that the base cope staining of the miR-224 show clearly that negative correlation of the ZIP4 protein with the miR-224 in tumor tissue. When overexpression miR-224 in pancreatic cancer cells, we found that it can reverse the ZIP4-induced cell migration phenotype. Furthermore, overexpression miR-224 could lead to increased apoptotic molecules caused by the UV irradiation. The similar trend was also observed in the tunnel staining positive cells. Next, we checked the miR-224 target gene, API5. We found that miR-224 mimics could inhibit API5 genes with terminal UTR reporter plasmid in pancreatic cancer cells, where abolishing this binding sites could rescue this inhibition effect. Furthermore, we found that knocking down API5 in cancer cells could lead to increased cleaved PARP apoptotic markers, while reintroduce API5 back will reverse this effect. The similar trend also found in the tunnel staining cells. Next, we further investigate how ZIP4 negative regulates miR-224 expression. We found that a negative transcription factor REB1 could bind to the miR-224 promoter region by chip PCR, and knocking down ZIP4 or REB1 could significantly increase the miR-224 promoter activities, while mutation of this binding site could abolish the REB1 induced inhibition effect on miR-224 promoter activity. When we knock down ZIP4 in pancreatic cancer cells, we found that the phosphorylated form of the CRAB was reduced, and the REB1 protein level was also be reduced. We know that CRAB is a positive transcriptional activator of REB1 genes. Well, this phospho-CRAB is a known substrate of phosphatase called calcineurin. When we knock down ZIP4 in pancreatic cancer cells, it will show significantly increased calcineurin activity, which lead to the degradation of the phospho-CRAB and less REB1 protein expression. So when we treat cell with the calcineurin inhibitor CN585, it will just induce REB1 expression in cancer cells, while the miR-224 expression was significantly inhibited. So this is our working hypothesis. So we start from the high zinc level caused by ZIP4, leading to the suppression of the calcineurin and the increased phospho-CRAB, which induces negative transcriptional active factor REB1 and suppress the expression of the miR-224 in pancreatic cells. So the end effect of this cascade is the increased miR-224 target gene API5 and resistant to the apoptosis, as well as increased migration ability and promoting pancreatic cancer tumor progression. So these results we reported here might provide an insight explaining the part of how ZIP4 modulates the anti-apoptosis ability of cancer cells by regulating the expression of the miR-224 through calcineurin, CRAB, and REB cascade. Thank you for your patience.